Today we'll be talking about the urea cycle. The ammonia produced as a result of the amino acid metabolism is very toxic to central nervous system. This ammonia needs to get converted into some other substance which is less toxic to our body. As I have mentioned here, this ammonia is very toxic to CNS. So this ammonia needs to get converted into urea, which is less toxic to our body. And the cycle occurs in the liver. And the cycle is called as the urea cycle. So the conversion of the ammonia, the very toxic material, into the urea, which is very least toxic to our body, inside the liver, constitutes the urea cycle. The urea cycle is a 5-step reaction. I have mentioned it here. The first step is not considered as a part of the urea cycle. You will find in many of the books that they don't actually connect the first reaction with the rest of 4 reactions. But remember, the product formed from the very first reaction is very important for the continuation of the rest of 4 reactions. And in the rest of 4 reactions, I have mentioned it here that they generate amino acids as intermediates. The main thing about these amino acids is that these amino acids have no codons coding for them. Otherwise, we know we have at least 61 codons which code for all the different types of amino acids, which in turn form proteins. The amino acids formed in the urea cycle have no codons coding for them. So we conclude from this that the amino acids produced as the intermediates of the urea cycle do not form proteins. The 5-step reaction of the urea cycle, the first 2 reactions of the urea cycle occur in the mitochondria, and the last 3 reactions occur in the cytosol or cytoplasm. The very first reaction of the urea cycle is, I have mentioned it here, the ammonia is reacted with the bicarbonate ion, or simply in the books you will find the carbon dioxide instead of bicarbonate ion. But I have written the bicarbonate ion in front of the carbon dioxide because carbon dioxide never exists inside our body in the form of CO2. The carbon dioxide exists in the form of bicarbonate ion. So the ammonia is condensed with the carbon dioxide in the presence of water. It's a hydration reaction, consuming 2 ATP molecules to form carbamyl phosphate. So the net equation of the first reaction is ammonia plus carbon dioxide in presence of water, and 2 ATP is being consumed to form carbamyl phosphate. The enzyme used is carbamyl phosphate synthetase, as shown here. Now the product of the very first reaction is taken into the second reaction, and it is reacted with the ornithine. Now you might be thinking where from the ornithine comes. The ornithine in the urea cycle comes from the 5th reaction, as here in the 5th reaction ornithine is generated. This ornithine is actually consumed in the second reaction, and this ornithine is condensed with the carbamyl phosphate to form citrulline, and the enzyme used is ornithine transcarbamylase. So this carbamyl phosphate plus ornithine to give citrulline is the secondary reaction of the urea cycle. Remember what actually is done. From the carbamyl phosphate, the carbonyl group is taken from the carbamyl phosphate and is added to the ornithine. Once again I am repeating it, the carbonyl group from carbonyl phosphate is shifted onto the ornithine. In the process, phosphate is kicked out. We form citrulline. So this is the product of the second reaction. Now this citrulline goes into the third reaction. Remember, as I have mentioned, the first 2 reactions occur in the mitochondria. So this citrulline will cross the mitochondrial membrane and will go and will find its way to the cytoplasm. In the cytoplasm, citrulline will find its company with the aspartate. So citrulline combines with the aspartate to form organosuccinate. Remember, this reaction is energy dependent. It consumes at least 2 ATP molecules. But here, if you can see, I have here mentioned 1 ATP molecule is converted into AMP plus pyrophosphate. You might be thinking that only 1 ATP is being consumed here. Remember, the pyrophosphate which is formed here is then converted into 2 inorganic phosphates. the hydrolysis of one pyrophosphate is equal to the hydrolysis of one ATP molecules. In reality, actually the one ATP molecule is consumed, but likely if we can see here, pyrophosphate is converted to two inorganic phosphates. So this conversion of pyrophosphate into organic phosphate is equal as the hydrolysis of one ATP molecules. So totally we have consumed two ATP molecules in a sense, and we form organosuccinate. The enzyme used is organosuccinate synthetase. Now this organosuccinate is taken to the fourth reaction. Organosuccinate is converted into arginine and fumarate here. The enzyme used is organosuccinase. Now if you can see here, this fumarate, it connects the urea cycle with some other processes as well, as I will discuss very soon. Now this arginine, which is the product of the fourth reaction, is taken into the fifth reaction, and the fifth reaction is also a hydration reaction. We add water and we form ornithine, and urea is also formed. So in the very fifth reaction, in the fifth reaction of the urea cycle, we actually get the urea, and ornithine is also formed. This ornithine is then reutilized in the second very reaction of the urea cycle to ensure the urea cycle takes place continuously. Now this urea, since we know the structure of the urea as I have mentioned it here, so this urea contains one NH2 group, second NH2 group, and it has C double bond O. Remember, in the urea, the first NH2 group comes from the ammonia, which we had taken here. The second NH2 group, it comes from the aspartate, which we had used here. The carbon of the ammonia comes from the carbon dioxide, which we had utilized here. The oxygen comes from the water, which we added here. So this is overall structure of the urea, and the composition of urea comes from the whole cycle of the urea cycle. Remember, the malate or the fumarate produced from here in the fourth reaction can be converted into malate. This is a reaction of the Krebs cycle. Then this malate, or simply, yes, it's a reaction of the Krebs cycle. Then this malate is converted into oxaloacetate. From oxaloacetate we can form glucose through a process known as gluconeogenesis. Or this oxaloacetate can combine again with the acetyl coenzyme A in the Krebs cycle to restart the Krebs cycle. So we see here that the urea cycle has a huge relation with other processes like gluconeogenesis and Krebs cycle. And the net equation of the urea cycle is, we have consumed one ammonia here, one bicarbonate ion here, aspartate here, 4 ATPs, 2 here and 1 here. But I have mentioned that one pyrophosphate is converted into 2 inorganic phosphates. It's equivalent to 1 ATP. So as a whole, 2 ATPs here as well. So the 4 ATPs. And we form urea, we form fumarate, 4 ADP and 4 inorganic phosphates. This is the general crux of the urea cycle. Thank you.